biology is life.
Hey guys, what is up? Today I'll be talking about Catechol-O-Methyltransferase or COMT. This is an enzyme in the human body. In this lecture, I'll be going through the function of COMT, the expression and location of COMT, as well as the mutations and polymorphisms that affect the COMT gene. Finally, I'll be giving a brief summary of everything in this lecture. Alright guys, let's get into the function of Catechol-O-Methyltransferase. So COMT's normal function is that it metabolizes catecholamines. These are the hormones and neurotransmitters that are produced by the adrenal glands on top of your kidneys. Some examples here include dopamine, epinephrine, and norepinephrine. COMT inactivates and thus metabolizes catecholamines through a process called methylation. I'll talk more about this later. So, in the human body, there are two variants of COMT. The first variant is membrane-bound COMT or MB-COMT. This is a longer variant found in the nervous system. The second variant is called soluble COMT or S-COMT. This variant is found in the bodily tissues. For those of you who want a more molecular-based explanation of COMT's function, I broke down COMT's function here into 3 simple steps. So in step 1, the catecholamines and the other substrates will bind to the COMT enzyme. This binding of enzyme and substrate is almost a universal process in all enzyme-driven reactions because the substrate have to bind at the enzyme's active site in order for the enzyme to lower the activation energy of the reactions. The second step is where the methylation of the catecholamine actually happens. So methylation is basically the process where a CH3 group is attached to a molecule. In this case, the molecule is dopamine. In this reaction, the methyl group donor is a molecule called S-adenosylmethionine or SAM. This is a common methyl group donor molecule in a lot of biological reactions. Finally, in step 3, the inactive metabolite as well as the other products of the reaction is released. Now let's take a step back. It turns out that Catechol-O-Methyltransferase actually interacts with a variety of different proteins when it's carrying out its tasks. In this lecture, I'll be going through 3 of these proteins. The first 2 are MAO-A and MAO-B, and the third one is DDC. So to give a brief background for these proteins, MAO-A and MAO-B are also called monoamine oxidase A and monoamine oxidase B respectively. They're also degradation proteins, which means that they can both catalyze the first step of catecholamine breakdown. I'll talk about this process more in depth later. For DDC, it's also known as dopa decarboxylase. It's a lyase enzyme and it synthesizes dopamine, which is a COMT substrate. Alright, so how does MAO-A and MAO-B interact with COMT? Well, as we've said before, monoamine oxidase A, monoamine oxidase B, and Catechol-O-Methyltransferase are all degradation enzymes. This means that they can work together to degrade catecholamines such as dopamine. As you can see here in the image on the left, dopamine is being degraded by both monoamine oxidase and Catechol-O-Methyltransferase. Notice that in the diagram, the order of enzyme metabolism does not necessarily matter. For example, you can see here that dopamine can be either broken down by monoamine oxidase and Catechol-O-Methyltransferase in the first step, only to have the opposite enzyme metabolize it further in the second step. For the image on the right, you can see that Catechol-O-Methyltransferase and monoamine oxidase are not the only enzymes that break down catecholamines. The metabolism of catecholamines is a very complex process which requires a lot more than just 2 enzymes. Now for dopa decarboxylase or DDC. As you can see here in this DDC function diagram, dopa decarboxylase actually synthesizes quite a bit of different molecules. However, the most important thing to take away from this diagram is that DDC synthesizes dopamine from a precursor molecule called L-dopa, and dopamine is a very important substrate of COMT. Basically, DDC makes a molecule that COMT breaks down. In order to get a more complete understanding of the interactions between COMT and different proteins, check out this string diagram. You can see here in this diagram that there are lines connecting proteins together. Each of these lines represents a study that has been conducted linking the 2 proteins' functions together. I've also circled the proteins that we went over in this lecture in red. This list of proteins obtained from the string database are the ones previously displayed in the string diagram. You can see here that I've also marked the proteins we went over in this lecture with a red marker. As for the numbers on the very right, these numbers represent the percentage for which the studies linking COMT to the specific proteins as shown in the previous diagram were not due to chance. In other words, the higher the number, the higher the chance that the specific protein shown is interacting in some way with COMT. Because of catechol-O-methyltransferase's important role in the degradation of catecholamines, it means that diseases that pertain to the concentration of catecholamines in the body will also be very much related to COMT. One of these diseases is Parkinson's disease. A major factor that causes the symptoms of Parkinson's disease is a lack of dopamine. As you may recall from before, catechol-O-methyltransferase metabolizes dopamine. This means that drugs that inhibit COMT activity will increase dopamine levels in neurotransmission and will mitigate some of the symptoms of Parkinson's. And this is why some Parkinson's patients take COMT inhibitors like entacapone along with their normal medication in order to lessen the symptoms of Parkinson's. So, COMT is basically present everywhere in the human body. But where is its predominant location in the central nervous system? In the bodily tissues? Where is its genetic location? Well, let's find out. This human protein atlas diagram shows the places where COMT is detected in the brain as well as the specific concentrations of COMT in these areas. In this diagram, I circled the regions with the highest concentration of COMT in a red marker. As you can see here in the image, the concentrations of COMT in areas of the forebrain, such as the cerebrum and the limbic system, are higher than the other regions. This is because these regions use more dopamine compared to other regions, which means that there will be a greater need for COMT to break down the catecholamines in these areas. Within the forebrain, COMT plays an exceptionally important role in the prefrontal cortex, shown here in the diagram. This is because there's less of a specific transporter called the presynaptic dopamine transporter in the forebrain compared to other regions. Like catechol-O-methyltransferase, the presynaptic dopamine transporter's function is to get rid of dopamine from the synaptic cleft, though it gets rid of dopamine using a different pathway called reuptake. Because of the decreased number of presynaptic dopamine transporters in the prefrontal cortex, it means that COMT will have to play a heightened role in that area. Now for the bodily tissues. As you can see here in this Manhattan plot, the epididymis, the testis, and the urinary bladder express the most COMT. I couldn't find any information for why this is the case, however. Even though organs like the epididymis, the testis, and the urinary bladder may express the most COMT, it doesn't mean COMT's function is necessarily restricted into those areas. For example, COMT is very abundant in the liver compared to other organs. For those of you who don't remember, the liver plays a role in the detox of chemicals as well as the metabolism of drugs. Because COMT's role is to metabolize catecholamines, soluble COMT will play a big part in the liver in assisting the breakdown of certain molecules. Now let's talk about the cellular location of COMT. As you can see here in the two diagrams that I've obtained from the Human Protein Atlas, you can see here that COMT can be found in the endoplasmic reticulum, the vesicles, and the cell membrane within the cell. It's also noted that COMT can be found outside the cell or extracellularly. Again, these regions are circled in red. Now, let's take a look at the COMT gene's chromosomal location. So, as you can see here in this diagram, the gene is located on the long arm of the 22nd chromosome, position 11.2. This is indicated by the notation 22q11.2. Speaking of chromosomes, let's talk about the single nucleotide polymorphisms and the mutations associated with the COMT gene. Obviously, there are too many different single nucleotide polymorphisms and mutations associated with the COMT gene to cover in this one lecture. Because of this, I'll only be going through the variants RS468 and RS4646312 for single nucleotide polymorphisms. As for mutations, I'll be going over the 22q11.2 deletion syndrome. Let's talk about single nucleotide polymorphisms. So, what exactly are single nucleotide polymorphisms? Well, a single nucleotide polymorphism is a substitution of a single nucleotide base within a specific part of the genome. You can say that substitution mutations and alleles of genes are basically the same thing as single nucleotide polymorphisms. However, different from substitution mutations, single nucleotide polymorphisms have to meet one more requirement. They have to be in at least 1% of the population. Take a look at this image of the COMT gene SNPs from the GWAS catalog. You can see here I've circled in red the SNPs that we'll be going over in this lecture. I've also boxed in red the symptoms and the reported traits associated with the SNPs that we'll be going over. Okay, let's talk about the RS4680 SNP, also called the Val158-MET SNP of the COMT gene. So this single nucleotide polymorphism is actually the most studied SNP of all of the ones associated with COMT. It involves a guanine that changes to adenine in the 158th codon, which changes a valine amino acid into methionine amino acid. In the wild type, which is basically the natural variation of the COMT gene, there are two guanines in the area which yield two valine amino acids and is associated with lower dopamine levels. There's a mixed genotype which has one adenine and one guanine, and that yields one valine and one methionine. This one is associated with intermediate dopamine levels. For the polymorphism genotype, there are two adenines and they yield methionine and methionine. This genotype is associated with higher dopamine levels. The reason why differing RS4680 variants are associated with different levels of dopamine is because the shape of the enzyme changes when methionine replaces the valine amino acid in the normal COMT enzyme. This resulting variant of COMT is only 25% as effective as a wild type. Because the Catechol-O-Methyltransferase resulting from the polymorphism is much less effective compared to the wild type, it means that dopamine in the individuals with the polymorphism is much less likely to be broken down. Thus, there's higher dopamine levels in individuals with the polymorphism. The higher dopamine levels in the individuals with the polymorphism also have consequences. For example, they may be more susceptible to diseases like schizophrenia. This second SNP I'll be talking about is the RS4646312 SNP. This SNP is a replacement of a thymine to a cytosine and is linked to schizophrenia, but the exact mechanisms of how this is the case is still unknown. Finally, for mutations in the COMT gene, check out the 22q11.2 deletion syndrome, also known as DiGeorge syndrome. As we said before, 22q11.2 is the precise chromosomal location for the COMT gene. DiGeorge's disease deletes 30 to 40 genes in this area, leaving only one copy of COMT in cells. This means that there will be abnormal regulation of catecholamines in the cells. This creates a whole host of behavioral problems as well as a risk in Parkinson's disease. It also must be noted that 90% of the cases are sporadic and only 10% are autosomal dominant. This suggests that this disease is not mainly inherited through genetical means but rather spurred on through outside factors. These outside factors are still unknown. Alright, now let's get into a very brief summary of COMT. So the main takeaway here is that COMT is an enzyme that metabolizes hormones in the body called catecholamines. And mutations and SNPs of COMT are associated with symptoms such as psychosis, schizophrenia, etc. You've now reached the end of the video. If you enjoyed the content, there's a little red button down below that says subscribe. Please click that and please also give me a like as well for the YouTube algorithm. Thanks guys, and I'll see you in the next one.